Hi everyone, this is lecture 33, more quantitative genetics. As we saw last time, most traits in real genetics don't have simple ratios like Mendel found. And so quantitative genetics is an attempt to understand how those distributions that we see in the real world come about. This material again is from chapter 22, page 650 to 655. First question, two average height parents have several children, and the children have a wide range of heights, with some taller than their parents and some shorter. Which set of parental genotypes could account for those data? Mark all correct answers. Number 2, in ryegrass, seed color is a polygenic trait. Additive model. If true breeding red and white varieties are crossed, the F1 are intermediate in color. If the F1 are self-fertilized, about 1 in 64 have white seeds. How many loci are segregating color alleles? What we saw last time was that when multiple genes are involved, and each showing incomplete dominance, the resulting ratios look more like a normal distribution. A normal distribution is one where the average phenotype is the most frequently seen, and the extremes are the rarest. The one we looked at last time was wheat. And in that case, wheat color, the red versus white, was controlled by two genes, let's say genes A and B. This is the example right here. Crossing two plants that were intermediate in color gave you a range of colors from pure red to pure white, where pink was the intermediate. The same trend is seen with other examples where multiple genes are included, except that the resulting normal distribution looks more and more gradual. With 3 genes, A, B, and C, you notice that the range extends even further and looks more like a normal distribution. 4 genes, A, B, C, and D, and A, B, C, D, and E. More and more phenotypes are seen in each of these cases. With normal independent assortment, again, ranges of phenotypes are seen. This is seen with, for example, the wheat, and for example, eye color. Here's an example where two people who are have an intermediate in color of their eye, sort of a hazel color, have these are the possible progeny that would be resulting from that cross. So the parents or genotypes are big A, big B, little b. The gametes that would be made are big A, big B, big A, little b, little a, big B, and little a, little b. And these are the ranges of colors that you would obtain from such a cross, from dark eyes to blue. And like we did with the wheat, you can show this as a graph where the average is the most frequently seen. Again, it's because it's the most ways that two additive alleles can be used in combination. For example, in this case, there are 6 different combinations that can give you 2 additive alleles, big A and big B, or 2 big Bs or 2 big As. The extremes have only one possible way that their genotypes could give you that phenotype. For example, dark eyes would only be resulting from 4 additive alleles, whereas the blue eyes would only result from no additive alleles. So some implications from quantitative genetics. Individuals with the same phenotype may actually have different genotypes, and we saw that with the eye color. Different genotypes could give the same eye color. For example, in this problem here, these genotypes all have 4 additive alleles. They would all have the same phenotype, and yet their genotypes are very different. This explains things from crops management to human health and disease. For example, some people may all have the same disease, for example, diabetes. And yet, the reason why they have diabetes could be very different. They may have different risk factors, and so that leads to different outcomes when being treated with medicines. Some medicines are more effective on certain genotypes than others, and so some medicines won't work for some diabetes patients as well as others, because their genotypes are very different, even though they all have the same phenotype. Crosses between 2 true breeding parents can produce intermediate F1 hybrids. That when those are crossed, may be able to produce F2s that are more extreme than the original parents. Here's an example. These two parents here, each of which have 4 additive alleles, have different ones. They produce F1 that have, again, 4 additive alleles, and yet crossing these F1 together would give you some that have the extremes. One of the questions we would like to know with quantitative genetics is approximately how many genes are contributing to a trait. This is important, again, for agriculture, for human health and medicine, and things of that nature. So how do we do this? Well, it's pretty hard in reality, but we can approximate it using a very simple formula. What we're going to be using is this formula here, 1/4 raised to the n, power of n, where n is the number of genes that are contributing to that trait. 1/4 to the n is equal to the ratio of the F2s expressing the extreme phenotype. Let's see an example. I'm going to go back to snapdragons for this example. Here's a simple case where snapdragon color is controlled by one gene, gene A. The additive allele, big A, is making the red color. The non-additive allele, little a, is making the white color. 1/4 to the n is equal to the ratio of F2 individuals expressing either extreme phenotype. You can count up the number of offspring in this cross, which is 4, 1 plus 2 plus 1, and you would set that ratio equal to 1/4 to the n. So, for example, the ratio of plants exhibiting an extreme phenotype would be 1/4, right? Because 1/4 of these plants are red. Or you could do equally with white. 1/4 of the plants are white. So 1/4 is equal to 1/4 raised to the n. And the math here is very simple. N is equal to 1. And we can see that the gene A is the only gene controlling color here. So again, the formula 1/4 raised to the n is equal to the ratio of F2 individuals expressing either extreme phenotype. Let's take a slightly more complicated example with wheat. Here we know that there's 2 genes, A and B, controlling wheat color. Again, 1/4 to the n is equal to the ratio of F2 individuals expressing either extreme phenotype. Looking at the extreme phenotype red, you can see that there's 1 red for every 16 total wheat that are produced. So the total F2 offspring are 16, but only 1 of those is red. So in this case, the formula 1/4 to the n is equal to 1/16, which is the fraction that are red. The math again here is pretty simple. You know that 16 is 4 squared, so n is equal to 2. 2 genes, A and B, that are controlling the color. Of course, the problems could get more complicated, so it's important to understand how that works. Here's an example where the distribution is a little bit more spread out. How many genes contribute to this phenotype? Well, you begin by adding up all the individuals. 1 plus 6 plus 15 plus 20 plus 15 plus 6 plus 1 should equal 64. One of those 64 is going to have the extreme phenotype. So 1/64 is set equal to 1/4 raised to the n. Now you may know that in this case 64 is 4 cubed, so that n is equal to 3. But if you wanted to solve for n using a math formula, you could do it this way. Just rearranging the formula by inverting both sides gives you 64 equals 4 raised to the n power. All I did was invert the fractions. To solve for the exponent, you need to take the logarithm of both sides. So log 64 is equal to n times log 4. Remember that when you take the logarithm of an exponent, the exponent comes out in front of the logarithmic function. Now it's simply a matter of solving for n. N is equal to log 64 divided by log 4. Using your calculator, you can find that n is equal to 3. Alright, that is all for today. I will see you in class.